Thank you.
Good morning. I love being introduced by Julianne, and I'm going to really miss that. We're going to miss you. So uh, I've been doing this for a few years now, and every year along about the middle of May, when it's time to turn in the list of the speakers, um, I have maybe three, and I go into my full panic mode. So this is the second or third year in a row that I have 7 speakers for 6 dates. So we will have 7 weeks of this. And the talks this week, this summer, will be going from the really tiny to the really big. So later on, the really tiny talk will be about antineutrinos. And then we have a talk later with about viruses, and then going up the scale today, we're going to have bacterium. And then we make a little bit of a leap and we go to the big toe. There'll be a talk about the big toe. And from there, we go to strawberries. And then the really big is going to be about the Red Sea and experiments and diving in the Red Sea and the results of that. So today, we're lucky to have Michael come and talk to us today. He is from South Florida, and he's going to tell us all about our favorite bug, E. coli. Michael Patrides, thank you for coming.
Hello, everyone. I just wanted to start out by thanking Walter and all of you for having me out here to do this talk, and thank you all for coming to see it. So just to give a really, kind of quick introduction here. Excuse me. My name is Matthew. I'm from Dr. Linda Bloom's lab at the UF Department of Biochemistry and Molecular Biology, and I'm going to be talking to you about some of the work that I've done as part of my PhD project the last year and a half or so. Which regards the dynamics of E. coli helicases and their role in the DNA damage response and repair mechanisms. Also, if at any point during this talk you can't hear me or if I can clarify something, feel free just to holler at me. All right, so I'm going to start just by giving some kind of brief, kind of an overview background here. So a lot of people may not be super familiar with this subject, so I'm just going to kind of give a general introduction here. So DNA damage is something that is really ubiquitous for every organism that is alive. It's something that's just pervasive. It's in every cell, every minute, every second of every day. It's something that's just constantly occurring. And it comes from a variety of sources and works by a variety of mechanisms. So you can see here some of these common sources which may induce DNA damage. So you have things like reactive oxygen species, which are these sort of toxic compounds in cells that can cause structural damage to the DNA. You can have replication errors, which are when the enzymes that are responsible for making our DNA just kind of make mistakes because nothing's perfect. You can also have exogenous agents like UV radiation or different drugs or chemicals, and these can cause different damage features in and of themselves. And so you have all these varieties of damage that here can happen, and you have a variety of different damages that result from these. So things like single strand breaks, which is where one part of the DNA backbone will have a break in it. You can have base damage, which is where one of these DNA bases will somehow be damaged or modified so that it's no longer functional. You can have mismatches, which is where the wrong base is incorporated, and so you don't have the proper DNA code at that point. And you can even have really bad things like double strand breaks. And this is where there's just a complete break in the DNA. And as you might guess, that's very bad. And so you have this variety of DNA damage that can occur. And so with all of this going on, it kind of raises the question, how is genomic integrity maintained? How are our cells still functioning and alive? And the answer to that is that there's a variety of DNA damage responses and repair mechanisms that are responsible for making sure these instances of damage don't end up killing our cells. And so I'm just going to do a really brief overview of some of these. So you have things like base excision repair, mismatch repair, and nucleotide excision repair, where basically you can take out specific bases from the DNA and then replace them with the proper bases, and then synthesis can go on and everyone's happy. You have some processes over here like homology directed repair, which basically uses other DNA in the cell kind of as a template if things go really wrong. And that's sort of a more error-prone mechanism, but it still works nonetheless. But basically, what you should just know is that there's a variety of processes here that work by different means, all towards the end goal of ensuring that our DNA is stable and its integrity is maintained. And then also something specifically to know is that DNA damage it can occur anywhere in the genome, but it commonly will occur at replication forks. And what I mean by replication forks are places in the DNA in a cell that's actively being replicated. So it's where you have one DNA that's being synthesized then to make two individual daughter strands of DNA. And this is something that happens in every cell. So it's a very common process that occurs. And there are different processes that can take over for a damage repair, or what we call tolerance in these instances, which basically is instead of repairing the damage, you just sort of tolerate it, and you can kind of skip over it and come back and fix it later so that the cell can continue living and just hopefully mitigate downstream risk at that point. But what you should know about a lot of these processes is that in many of these repair and tolerance pathways, there are these specific proteins called helicases that are required. And helicases are enzymes that require the use of ATP, which is basically just an energy source. And what they do is they will move along DNA and they will separate double-stranded duplex DNA into its constituent two single strands. So basically, it's just kind of a motor that runs along the DNA and just splits it apart. And that's necessary because to be able to fix the issues with DNA, you have to be able to access the bases that are inside the backbone. And so these are necessary to kind of open it up for that repair. And so kind of what my lab looks at specifically are helicases in this one certain family of enzymes. This family is called the XBD Rad3 family, which is just named after this helicase right here and another one, Rad3. But what you should know about this family is that they're in all domains of life. So it's from humans to bacteria to yeast, everything has these. So they're really quite pervasive. You have 4 family members of these helicases that are in humans, and all of them are very important for maintaining genomic stability and repairing our DNA. And so consequently, if you have issues with these 4 proteins, so maybe they're having some kind of dysfunction or they're not being made properly, you have resulting diseases that can develop because of that. So for instance, you might have things like xeroderma pigmentosum or Cockayne syndrome or Fanconi anemia. And these are all diseases that have been pretty well characterized, and we know that they're directly a result of issues with these 4 human helicases. So basically, these enzymes are very important in maintaining genomic stability. And so what my lab kind of looks at in particular, I just talked briefly about the human helicases in this family. We look at the versions of these helicases that are in bacteria, specifically E. coli. And so before I kind of get into what I work on a lot, I should address like kind of before a lot of the work that I've done and my lab has done, there really was only one known family member of XBD Rad3 helicases in E. coli, and this enzyme is called DING. At this point, DING has been really quite well studied, and we understand pretty well how it works, what it does, and what its role is. And this diagram here is just meant to show that here in humans, you have these few helicases, FANCJ, RTEL1, et cetera. And then kind of in parallel, you have DING and E. coli. And so DING, we know at this point resolves replication transcription collisions. And this is basically when you have on the same piece of DNA, replication machinery and transcription machinery, and they kind of just move towards each other and eventually they're going to hit. And so at that point, something has to give. And so what happens is we can get DING in there and it will actually resolve those and make it so that the genome is not compromised in those instances. And so that's just kind of a brief background on these helicases in E. coli specifically. So to get into what my project entails more of is more than a few years ago now. But we have collaborators in the lab of Dr. Susan Lovett, who's over at Brandeis. And they made this interesting observation where basically in genetic screenings of E. coli, they saw that the cells were tolerant to the drug AZT in low doses. Now, AZT is a drug, which is called a thymidine analog, which basically means it just looks like thymidine, which is a naturally occurring DNA base. It's in all of our DNA. It's in all of everything's DNA. But the thing is, it's slightly different. So you can see thymidine has this one little OH group at the bottom left here, whereas AZT has this 3 nitrogen group, which is called an azide group. And the thing is, if cells incorporate AZT into DNA instead of thymidine, which is easy because they look similar, it makes it so the DNA can no longer be extended, and it will cause replication stalling, which is very bad for the cell. And that's kind of what this looks like here. So you can see this green enzyme is a polymerase, which is synthesizing new DNA. You have this kind of red stop sign looking molecule, which is AZT. If that's mistakenly incorporated into DNA, the polymerase can no longer keep making DNA, and it will just get off and leave this stalled fork right here, which basically is just a kind of a dead end for the cell. It can no longer replicate that DNA, which is, as you might think, very bad. So it was very interesting that our collaborators turned up that there are these 2 genes in E. coli, YOA and Hal-C, which confer tolerance to this drug, which basically means that when you introduce this drug to the E. coli, as long as these 2 genes are intact, the E. coli can survive and tolerate this drug. And so that makes us wonder, how is this going on? Like, what kind of repair mechanism is involved to excise this drug from the bacteria's DNA? And so that's kind of where my project started to begin here, and what I'm going to be talking about today. So as I mentioned, there were those 2 genes that are tied to the ability to tolerate the drug AZT. One of those genes is YOA, and this gene encodes the protein called YOA, which is very straightforward. But YOA is a 5' to 3' translocating helicase, which is in E. coli. And when I say 5' to 3', I just mean that it's going one specific direction along DNA, because DNA is it has what we call a polarity to it, so it's not perfectly symmetrical. So it will go specifically one direction and not the other direction, which is kind of key when describing these enzymes. But what you really should know is just that YOA is a helicase. So it's like I was saying, it'll just get onto DNA and it will split double-stranded DNA apart. So you just have 2 single strands instead. And YOA is part of the XBD Rad3 family, which I was talking about earlier. It's closely related to DNG, which is the one that I had said was previously described well in E. coli. It was the only other member of the family in the bacteria. And originally it was identified as a helicase based on sequence similarities with DNG. It shares about 29% of its amino acids with it, and it has many of the same common structural motifs that are found in these helicases. And this is actually just kind of a predicted structural model to the right here. So we do not have an actual structure of this protein yet. We're working on that with someone in the lab right now. But this is a predicted model from software called AlphaFold, which basically uses the protein sequence to predict its structure. And you can see, like, well, maybe not, but there are different structural motifs here that are really what we would expect to see in a helicase. So we feel fairly confident that this structure is reliable. All right, so now I'm going to go over kind of the other piece to this puzzle here. I mentioned again that there were 2 genes earlier, YOA and Hal-C. This is the other gene, which is Hal-C. It encodes the protein called chi, which is just denoted by this Greek letter here. So chi is interesting because this had actually previously been studied quite a lot, but in a completely different context. So this protein is a small subunit of what we call the DNA polymerase 3 holoenzyme, which is a large term that basically just means this whole complex that's responsible for replicating DNA in E. coli. And chi is part of this. You can see it's this little red circular part here that's right near the bottom. And chi has been well characterized in this role. Basically, it's responsible for connecting the rest of the polymerase 3 with this other substance called SSB, which is a single-stranded DNA binding protein. And the whole purpose of it here is basically just to increase the efficiency of the polymerase as it goes through the genome. But it's not too important to know that right now. And so now that we've kind of gone over those proteins individually, I'm going to be talking for the rest of the day about this kind of combined protein complex, which is called YOA chi, which is essentially just those 2 proteins bound together. And so we know this is a complex because we've done a lot of biochemical work in my lab, me and other graduate students. And we know that chi can actually bind to YOA instead of binding with the DNA polymerase 3 in E. coli. And so it forms this specific complex that when the two proteins are combined, it acts as a functional helicase instead of just having the two separately. So for the rest of the talk, I'll be referring to it as Yoa-Kai because that is the functional form of the helicase when those two are combined. All right. And so now I'm going to start getting into some of the data that I've collected here, which is going to regard basically the interactions of this helicase, Yoa-Kai, with a different protein that I mentioned a bit earlier called SSB. And we're going to be looking specifically at this at the single molecule level, which is actually something that's pretty new for our lab and also the field in general. It's actually this really kind of cool technique that I'll sort of describe to you in a minute, but it's very unique from anything we've had access to in the past, and it's very exciting. So I'm going to give a brief rundown of this other protein called SSB. Again, it's just single-stranded DNA binding protein. So basically, this is a protein that when you have exposed single-stranded DNA in the cell, which can happen in instances of DNA damage or even just routine replication, SSB will very tightly and very readily bind that single-stranded DNA because you want to protect it since it's already kind of in a easily compromised state. And so that's kind of its job in most pathways is it'll just protect single-stranded DNA. And it does that by actually having the DNA wrap around the protein, which is interesting. It's not like it just contacts it and binds it. The DNA will physically wrap around the whole protein. But the interesting thing about this is that SSB can actually interact with other proteins that are also on DNA to exert different kinds of activities that might not be doable otherwise. And so we have here kind of a just general representation of what my lab thinks is happening in E. coli to resolve the AZT incorporation damage, which I described earlier. So what we think, just kind of very in brief, is that at a stalled replication fork, which is represented by this red X, which you can take as meaning AZT, because that means the DNA replication is stalled and it can no longer be extended. We have SSB, which binds the single-stranded DNA. And then you have polymerase 3, which, as I said, can no longer make new DNA because of the drug. What we think then happens is that Yoa-Kai will come in, interact with this SSB, and it'll actually chew up some of the DNA and split it apart, which you can see here, and it'll expose that damage so that another different enzyme, like an exonuclease, can come in, digest it, and then it'll be gone so that the polymerase can come back in and keep replicating as if the error never happened in the first place. So this is still very much a work in progress, but we think this is what's going on here is that Yoa-Kai, which is this helicase, has interactions with the SSB protein. All right, and so now I'm going to give you kind of a really brief explanation of what I'm going to be showing you in the next few slides. So the apparatus that we use to do these single molecule based experiments is called a C-trap. And the general type of instrument that it is what we call optical tweezers. And so how this machine works is that you actually have two very powerful lasers that are projected into the machine. And these lasers can actually suspend very small particles within the lasers. And you can then manipulate those small particles in the machine. And so what we do is we use those lasers to suspend very small polystyrene beads, which you can kind of see here. And those beads are coated in special chemicals that allow us to interact with different molecules. So in this case, what we're going to interact with is DNA. And so what I'll do in these experiments is I'll take these beads suspended in the lasers, and I can actually stretch a single piece of DNA between those two beads and then manipulate that one strand of DNA, which historically has not really been possible through any other techniques that we've had at our disposal. So this is very exciting. And so just to kind of show you a little bit about how this might look. You have kind of represented here two of these beads, which are in these powerful lasers I was talking about, and then a strand of DNA, which is suspended between these two beads. And so what we can do at that point is we can actually fluorescently label proteins that we're interested in. And then when those proteins bind on the DNA, we can actually take little pictures of those. And we can take scans to see what the proteins are doing on one strand of DNA, which is very interesting because we have never been able to see what individual proteins might do on DNA. We just do assays that we look at thousands or millions of molecules at the same time. So this is very interesting. And so practically what this might look like is you see over here, you have this green molecule. It's kind of moving back and forth on the DNA. You can see it represented over time here by this kind of adjustment of the green fluorescent signal. And as it moves down, it'll kind of trace that out in what's called a chymograph. And these chymographs are, you can kind of think of them as molecular movies, where you're sort of tracing the position of a fluorescent protein over time, and you're putting all those little pictures together to make a movie. So here's kind of what that might look like. So this is kind of a graphical representation of what a substrate I'm dealing with here. So we have these two beads that I've been referring to, and in between them, I've suspended this one piece of DNA that has three kind of sections to it. The top section is double-stranded, the middle is single-stranded, and the bottom is also double-stranded. And so what we can do is bind different proteins to each of these sections, because different proteins will have different affinities for either double or single-stranded DNA. And so what we do is in the double-stranded portion, we have this green fluorescent molecule, and in the middle, we have a red fluorescent molecule. And this is a real picture that I collected from our instrument, where you can see each of the two large beads. And in between them, you just have this line here that's colored. And that's the DNA itself, actually. So you can see that part of the DNA is green, then red, and then green. And that's directly representing what we see here in this schematic. So what you're looking at is just an actual single strand of DNA represented by that fluorescence. And so what we can do is then take pictures of that same strand of DNA every 50 milliseconds or so. And when we line up all those pictures, we get a chymograph here. So you're basically just seeing the same single strand of DNA just every 50 seconds or 50 milliseconds right in a line. And so it makes this kind of movie that you'll see as time goes on. And so to sort of talk about kind of things I've looked at on this. So we work with the helicase Yoei-Kai, as I've been describing. And what we have seen with this substrate, which again is where we have this single-stranded gap, we can actually put our protein on the middle section here, which is single-stranded, and we can actually watch it unwind the double-stranded DNA. And we can do that because, as I said in the previous slide, we have this green molecule that we can bind to the double-stranded DNA. And so as our helicase unwinds and there's less and less double-stranded DNA progressively, you can actually see that this green section on top will slowly just kind of taper off and as the black area kind of expands. And so this is actually a direct measurement of us seeing less and less double-stranded DNA as our helicase is unwinding it. So this is really cool because we're actually seeing our helicase just do its job on an individual piece of DNA and unwind this. And so then I'll kind of go briefly over this as well. So this is another cool project that I've been working on, which is where we have our helicase, which, as I said, we have that other protein called SSB, or single-stranded binding protein. And we want to look at the interactions of that helicase with SSB. And so what I'm looking at here is I have suspended between the two beads in our tweezers just single-stranded DNA. There's no double-stranded DNA here at all. And the enzyme, the helicase Yoei-Kai, is able to move along that single-stranded DNA. And so what we can see here is I have SSB that's actually been labeled so that we can see it in red, which is represented kind of in the schematic here. But in the chymograph over here, you can see that over time, we have all of these red lines that are kind of slanted. And basically what we believe this means is that the helicase is traveling with SSB, because they're all traveling in the same direction, which is exactly mirrors what we would expect from our helicase, because it exclusively moves in one direction on DNA. And the SSB itself is not a motor protein, so it's impossible for it to move on its own. But that's what we think is going on here, is that we're seeing the helicase drag along this other protein with it, which is very cool. And then we actually used a mutant that we made of this helicase, which I'm just calling R128A, which basically means we went in and we took this one critical amino acid residue and we mutated it to a different one. So we no longer can have the interaction between the helicase and the SSB. And then we did the same experiment to see what the differences were. And you can see that this chymograph here looks very different from that. And a lot of these red lines are much shorter and they're no longer slanted. And so what we got from this is that we think our helicase is actually specifically pulling the SSB rather than pushing it around necessarily. Because the only way that doing this mutation would then reduce the amounts of SSB interactions we see is if it were on the back end of the helicase as it's traveling. So what we're getting from this is that the helicase is pulling the SSB specifically rather than interacting with it some other way. We also verified that by doing just a negative control here, which is just SSB with no helicase at all. And you can see that this looks pretty similar to this, which again backs up the idea that the helicase is pulling along SSB behind it. And this is just kind of a quantification that I did, where I did a lot of runs of this experiment. And we see that there's a vast difference when I include the helicase, Yoa-Kai with SSB compared to both SSB alone and the mutant protein that we made. And you see it reflected in these numbers here. There's significantly more in the case of the helicase than in either the top or bottom case there. This is another kind of just fun project that I've just sort of started up. Here's what we're seeing, kind of the same thing in the past few previous slides, where we have the similar kind of slanted red lines of SSB traveling along DNA with the helicase. But in this case, we're actually exerting a force on the DNA of 30 piconewtons. And we do this because in our instruments, the DNA is connected by these two beads. So what we can do is actually drag one bead out a little bit, and this will stretch the DNA, and it will exert a set amount of tension that we can input into the machine on the DNA. And so we're doing this specifically at 30 piconewtons, which is quite high force for these proteins. And we can see something interesting that with Yoa-Kai, which is the helicase we work on, we still get these SSB tracks where SSB is traveling with the helicase. But with a different helicase, DIN-G, which is the other one that's in the same family as Yoa-Kai, we do not get those traces when we're at high force, like in this experiment, but we only see it when we move back to low force. And so essentially what this all tells us is that we think there's something really kind of unique going on with our helicase Yoa-Kai and how it interacts with SSB, something that which is not seen with other similar proteins and that might not have actually been observed before at all. So we're really excited about this, and we're going to eagerly continue investigating that. And so just to kind of give you a really quick wrap up of what I've talked about so far, as we saw that SSB, which are all those red lines on the chimerographs, we saw that it can move with the helicase Yoa-Kai, and that's because it's being pulled by it. And this fact, along with other structural data that my lab has obtained, as well as other predictions and studies in the literature, it indicates that our helicase pulls SSB specifically, and this is actually the very first observation to our knowledge of a mechanoenzyme like a helicase pulling a single-stranded DNA binding protein, which is really interesting because this could have some implications in how DNA damage repair processes work, because this is something we've never seen before. And so this could indicate that Yoa-Kai actually pulls SSB at stalled replication forks, which could play a role in how it helps resolve AZT-based DNA damage in cells. So that's something that I'm currently still chasing down, and we're really curious about how that might play into that mechanism. All right, and for kind of the second part of my talk here, I'm going to go into another sort of project that I'll change gears on a little bit. So I'm still going to look at the same helicase, Yoa-Kai, but we're going to look at it on these kind of weird secondary structures in DNA called G quadruplexes. And I'll kind of get into a little bit of detail about what that is first. So G quadruplexes, or as I might abbreviate it, G4s, are things that can hinder DNA replication in the genome. And so this happens because you can see here, this whole schematic is a representation of different types of DNA damage or different objects that can hinder replication. So you don't have to concern yourself with most of it, but this one location right here, these difficult to replicate regions, these are G quadruplexes. So you can see there's kind of weird looking 3D molecules that occur on the DNA. And so as you might suspect, that can block enzymes that are moving on the DNA, like polymerases, which are responsible for making new DNA. And so we're curious about how our helicase might interact with these structures and unwind them to allow for replication to continue on the DNA. And this is kind of what a G quadruplex looks like here. So it happens when you have DNA sequences that incorporate a few different kind of segments of guanine residues separated by other bases that it doesn't necessarily matter what they are, but you just have to have these kind of sequences of Gs that are interspersed with each other. And when you put into solution a metal ion like potassium, you actually get these sequences that can fold up in 3D space and make these kinds of bulky secondary structures. So you have the DNA that's kind of folding up and down and back on itself, which makes this weird looking structure here. And what happens is you have this kind of unique bonding pattern between the guanine bases called Hoogsteen base pairing. And what this allows us to happen is for this non-canonical structure that we would never normally see, can actually be stabilized and remain a structure in the DNA, which, as I said, is a problem for replication. And so the experiments that I've done to kind of start investigating this rely on a technique called FRET. And so this is fluorescence resonance energy transfer. And so what this technique kind of revolves around is we have these chemicals called fluorophores, which are basically molecules that when you excite them with a very certain wavelength of light, they will then emit a different wavelength of light. But something interesting is that when you put fluorophores really close to each other in physical space, instead of emitting a wavelength, a fluorophore can actually transmit its energy to a different fluorophore instead of emitting light. And so this is something that we can take advantage of for our experiments. So what I'm doing here is I have a specific DNA substrate that I use, and these red and blue shapes are representing the fluorophores on this DNA. And since they're very close to each other at the ends of this DNA, we're going to see PRET, which basically means that when I excite the blue fluorophore, I will not observe a wavelength coming out of it because it's transferring its energy to the red fluorophore. However, on the substrate, I'm going to put my helicase, and I'm going to let it unwind through G quadruplex and unwind this part of the DNA. So that way, when the substrate is separated, you get the red and the blue fluorophores that are no longer close to each other. And so in that case, when I excite the blue fluorophore, I can then see its distinct wavelength of light that is emitted instead. And so that's what I'm going to be looking at kind of in the next couple slides, is an increase in fluorescence, which correlates to an increase in unwinding, is what you can essentially think of this as. And so that's what we're looking at in these graphs here. So let's look at the blue curve first. So the blue curve is Yoei Kai, which is the helicase I've been referring to previously in the talk. You can see that there is over time an increase in fluorescence before it kind of levels out. And so this indicates to me that it is unwinding these DNA substrates with G quadruplexes in them. I compared this with DIN-G, which is the other closely related enzyme in the same family, also in E. coli. And that's in this purple curve, which you can barely see actually because it's so fast. But Yoei Kai is doing this much slower than DIN-G, but it's doing it to about the same level. So this is really interesting because it points to the possibility that Yoei Kai may unwind G quadruplexes in E. coli, which can help replication proceed when it otherwise would not be able to. And so I tried different conditions to kind of figure out what exactly was going on here. So I made reference before very briefly that potassium is something that we can use to cause G quadruplexes to form. So I tried different concentrations of that. I increased it from 1 to 70 to 100 millimolar concentration. And when I did those higher concentrations in the dark blue and the light blue, you can see those curves are much lower than the purple. And so what this says to me is that the increased amount of KCl of potassium is increasing the G quadruplex stability to the point where it cannot be reliably unwound by the enzyme anymore. And this could also have implications of what is going on in the bacteria itself, but it's still something that I'm kind of chasing down and trying to figure out. And just to kind of check myself, I tried these same kind of conditions ranging from 1 to 100 millimolar KCl just on different DNA substrate that I know Yoei Kai unwinds really well. And you can see these curves are all more or less the same, which tells me that the potassium itself is not actually influencing the enzyme activity just inherently. It really is something about the G quadruplexes that are determining the differences in unwinding that we see here. And so this is kind of something that's still more in its infancy for me, and I'm still kind of beginning to explore this. But what you can kind of take away from this is that Yoei Kai can unwind G quadruplexes, which are those really 3D bulky secondary structures on DNA, to a similar degree compared to DIN-G, which is that related enzyme, which is also in E. coli. I also saw that increasing potassium concentration will decrease unwinding activity of Yoei Kai on these substrates. and also that Yoakai is not affected by potassium on standard DNA duplexes. So what this likely tells me is that, as we've seen previously, AZT can cause stalled replication forks, which Yoakai helps to resolve, and it helps replication proceed. This may be another role that is helping replication proceed, because if you have polymerase that's replicating actively and it hits a G quadruplex, it may just stop and not be able to go on. And if you can get Yoakai helicase in there, it can unwind that so that the replication can then just proceed right through there and the cell is going to be fine. So I think this is likely another role that we're seeing for this helicase here that we have not uncovered previously. All right, so I'm just going to do a quick kind of wrap up slide here. I know this has been kind of a dense talk. So I'm just going to give you some quick take home messages here. So the helicase Yoakai, I've seen interacts with and pulls that protein SSB on single stranded DNA. And we believe that this kind of never before seen phenomenon probably relates to the mechanism of how the helicase resolves AZT damage, probably by pulling this protein away from the forks so that other proteins can possibly get in there to help chew up the damage so that replication can proceed. We also have seen that Yoakai will unwind G quadruplex structures as those can hinder replication. And so the bacteria can then continue to replicate through those as well. And as far as some future directions, as I've been saying, there is still a lot of work that needs to be done to exactly understand how the Yoakai SSB interaction helps to resolve AZT damage. We have a pretty good picture of it now, but there's still some things that we need to know to figure out exactly how this works. And the G quadruplex experiments are things I'm actually going to also start doing on our optical tweezers so that I can see what's happening on single strands of DNA and determine how the helicase is moving through those structures and if there's might be anything interesting going on there as well. So that'll be kind of the end of my talk. I just wanted to give a quick thank you to everyone in my lab. Savannah especially was a big help with a lot of the optical tweezer experiments. This is my committee members, and this is a nice little lab picture we have. With that, I'll have any questions that you guys might have or anything that you might want to ask about. And thank you for your attention.
I'm carrying a microphone so I can get questions from over here, but while I have it, I'll ask the first question. What does it all mean to me? Can I go in your lab and play with that optical tweezer?
We would love to have you. Yeah, so it is a lot of this is very kind of technical, I know, and it can be kind of tough to understand what the sort of end goal of this and how this relates to just everyday people might be. So a lot of what we're going for here is that we have this specific family of helicases that I kind of talked about in the beginning called XBD-Rad3 helicases. And the one that our lab works with, Yoakai, is one of those. And also there are those 4 helicases that are in humans that are part of the same family. So what we're trying to figure out here is that by understanding more of this helicase in E. coli, which is a very easy model system to work with, we hopefully can kind of parallel that knowledge with the human versions of these helicases to understand more of what their roles might be in the development of human diseases.
So how big is your optical tweezer machine?
It's about the size of that piano, actually. It's it's quite large, very expensive, actually. But
I know that a few years ago in our Nobel Prize series talks, a Nobel Prize a few years ago was for the development of the optical tweezer. So it's fantastic to see how it's being used. Some strange device that was hard to understand has now got something that can help you guys study things. Okay, questions? Were you waving your hand?
This is kind of a strange question, but AZT, it's a very familiar sounding thing. Is it a drug that's used or what is it? And then once you've told me what AZT is, could you connect it back to what you've just described?
Yeah, so the context that most people have heard AZT in is that's actually a drug that was used to treat HIV a lot of the time. So that's where most people might know it from, at least who are not like actively in our field. And so I'm not exactly sure about the mechanism of how it prevents viral replication with HIV. I would imagine it's somewhat similar to what we see with bacteria, with how it will cause misincorporation into the DNA and prevent replication, because then the virus would not be able to proliferate and spread. That would be my guess, but I wouldn't know exactly about how that mechanism works. But that's most likely where you've heard it, though.
My question was the same. What is
Yeah.
AZT? So it's not something that's around and it's a pollutant or something that we come across normally. It's a drug made for a specific purpose.
Yes. So AZT is it's specifically designed by us, and we can treat cells with it, or as I was just saying, we can use it as a drug for HIV treatment. What we're and I didn't cover this too much. Kind of what the purpose of what we're using it for here is it's something that we can very reliably use just to stall DNA replication, which can mimic a lot of pollutants can do that. A lot of various chemicals can be incorporated into DNA and stop replication from happening. And so we're kind of using it as a proxy for all of those types of damage that might be occurring, just because we know this is a system that's very
Yes,
easy.
it's a tool for you. It's a tool that
Yes.
you're using.
Yeah, it's a tool.
Thank you.
But it suggests that that AZT does also damage to while it's reducing replication of viruses, it's also damaging cells.
You're referring to like human cells?
Uh-huh.
Yeah. So again, I don't want to give you bad information here. I'm not an expert on that. But from what I understand, AZT, when it's used to treat HIV infections, actually does not really have too many off-target effects regarding human cells. I believe it can cause side effects, but not to the extent of just killing human cells, really, because it will, as I said, kind of prevent the spread of HIV infection, and it will help lower viral replication rates. But I don't think that it has the same effect for human cells. I'm not exactly sure about the mechanism, though. I would have to get back to you on that.
I want to make sure or ask you about something I'm not sure I understand. When you were talking about tolerance, that they're able to deal with it. Now, are they able to deal with it? Because it's going to build up. I mean, you're tolerant of this, and then you'll have that, and then that. Is it just a mechanism to deal with it immediately, and then it will go back and repair it? Is that what I'm thinking? Is that correct?
So that's a good question. When I'm speaking of tolerance mechanisms, there's kind of there's a couple more specific ways that can proceed. So some of what you said can be the case for some mechanisms, where basically if you have damage that's incorporated, you can have these special kind of polymerases that are called translesion polymerases. And so what they'll actually do is, instead of getting stalled at the damage site, they can actually polymerize over the damage and keep going. The thing is, though, you still have that damage there. And also, these polymerases are highly error prone. So when they keep incorporating DNA bases, they'll probably be putting in some of the wrong ones. So in the end, it can be kind of a negative in some cases. So you do have to then at some point get back with proper repair mechanisms and really go through those segments. When you end up with those kinds of tolerance mechanisms is when it's basically when the cell is desperate, like when there's really bad damage going on and it needs to just basically not die. It will use those kinds of error prone, really tolerance mechanisms just to keep surviving and then go back and fix the damage later. And there are some other mechanisms as well, which will work slightly differently, but that's kind of, I would say, the overarching theme for those tolerance mechanisms. It'll just kind of ignore it for a minute and then just kind of come back later and try to fix it when it's able to.
Any other questions? Put the last slide up, please. If you look down the left side under Savannah is Mitch Gulkis. Mitch spoke to us a year or two ago, and I've been lucky in getting past speakers to twist arms of future speakers. So I invite you to twist arms next year and help me get my 6 people and not have too much of an overflow.
Yes.
If there are no more questions, thank you very much. Wait a minute, I'll take a walk.
Is AZT azithromycin, which is sometimes used to combat bacterial infections?
No, so it's a very similar name, but that's actually a different drug. So azithromycin is an antibiotic that is used to treat bacteria. AZT is its full name is azithromycin. So it is very similar sounding, but it's a separate drug which works by a different mechanism. Azithromycin, I think, works by inhibiting bacterial transcription, I think, which is a different cellular process, but kind of the same end goal of basically preventing the cell from replicating. So different drug, same kind of results.
Thank you.
Okay, next week we're going to something slightly larger. A young lady will come and speak to us about strawberry variations and field work to make better strawberries. So we're going from really little to really big. I hope she brings samples. Anything further, anything else? Okay, thank you very much for coming and talking.
Thank you for having me.